this is kind of a sort of aside from the actual work that you're doing.
Yeah.
So the actual work that you're doing now in relation to FP, do you want to describe to us what you do?
I study something called, let's see, the major histocompatibility complex, or MHC. And basically, every vertebrate from turtle to human have these MHC proteins. And there's a couple different varieties of MHC proteins, but they all generally follow the following function. They are proteins that are expressed on the outside of cells, and they are really important for mediating how the immune system either recognizes its, self, the body, or invaders. So you can see here in these antigen binding cleft regions, basically there, if you have a perfectly normal cell, you're going to have MHC holding a little bit of peptide from something that was produced in that cell, say an enzyme. Then you're going to have immune system cells that come by and they might read what that MHC molecule or protein is holding, and it will be like, okay, everything checks out here. This is a protein that belongs to the cell. It checks out as self, carry on. Now, when a cell gets infected, say by a virus, you might remember from biology that viruses are like just on the cusp of being considered life. They can't really do much for themselves. So when they infect a cell, they actually commandeer the machinery in that cell to start producing the proteins that they need to survive. So some of those proteins, though, still get broken up, and pieces of them get expressed in this MHC binding cleft. Now, when an immune system cell comes by and it reads what that MHC molecule is holding and recognizes it as a foreign invader, it sets off basically a bunch of alarms within the body, and that cell is usually killed off by immune system cells. So the MHC is really that mediator of what's going on in the cell, and then letting the immune system know what's going on in that cell. The really cool thing about MHC, though, is that it's really polymorphic, meaning that in one given population, there might be multiple different MHC alleles. And generally, how diverse the MHC alleles are in a population is a good metric for understanding how susceptible or how immunocompetent that population might be in defense of a pathogen. So when we're talking about turtles specifically, as I said, immune systems haven't really been well studied in reptiles. It's been mostly in, mammalian model organisms. What I'm interested in knowing is how diverse are the genes in sea turtles that code for these ultra important proteins. And I was afforded the really wonderful opportunity to begin studying this in a really special group of sea turtles in the Indian River Lagoon. And what's really cool about these sea turtles is we go out and we catch them. And we've been doing this since 1982, actually. So here's kind of what a typical day out on the water looks like. This is a team deploying this large mesh tangle net. This tangle net gets soaked for 3 hours and it's continually checked. We are on this boat and we basically run the net hand over hand. And when we find a turtle entangled in this net, we use a large dip net, we scoop them up, and we bring them back to the workup boat. And that is where we take all sorts of measurements and samples, including skin and blood, and tumor tissue if the turtle has papillomas. So basically, we have a bunch of freezers that look like this. We have this extraordinary backlog of samples going all the way back even to the 90s.
That's a lot of samples.
A lot of samples. I mean, here's some blood samples you can see here. And I mean, we have so many that, it's really going to take a while for us to kind of really delve into them. But what's really great is they afford this really awesome opportunity to look at samples across time. So I've actually been looking at sequencing and then characterizing MHC genes in green sea turtles and in loggerhead sea turtles that have existed in the lagoon. And the one other thing that's really interesting about this is in the Indian River Lagoon in Florida, the sea turtles that live there aren't a genetic population. So there are genetic populations of turtles that all aggregate in the same area. So you have nesting mothers coming back to beaches in Florida and Brazil and off the west coast of Africa that all have the same genetic signature. But the turtles that are in the Indian River Lagoon, since they're juveniles, they represent sea turtles that have come from all over the Atlantic Basin. They are of genetic stock from all over the Atlantic Basin. So not only do I have a really nice temporal cross-section from the 90s to present, I also have a really nice cross section of multiple sea turtle populations, and it's kind of like getting a pulse check of, okay, how diverse are the MHC in this grouping, this aggregation of turtles, and can we relate it to whether or not a turtle gets FP?
Oh, that's really interesting. Well, in terms of like the diversity of MHC1, what do you think the allelic diversity might be, considering that these are endangered populations? So the population size is going to be pretty small. And so when you see small population sizes, you generally expect to see a decrease in the amount of genetic diversity. Do you think that's going to play out when you look at the allelic diversity in MHC1?
Potentially. So we have the results back from these juvenile turtles, and we're seeing that there's actually quite a lot of diversity in both rings and loggerhead sea turtles. But you bring up a good point, and this kind of goes back to the fact that this is not a genetic population. This represents input from lots of populations. So when we look at an input like this, it might be that, you know, the Indian River Lagoon and foraging locations like this represent, you know, the meeting of lots of different MHC alleles and strains. But the next thing that I want to do is I want to go and look at the MHC diversity in just a single nesting population, such as that along the Florida coast or even just beaches that we monitor, because it is possible that they will be much less diverse at those rookeries, at those breeding areas, than they are in the Indian River Lagoon or another area where you had a lot of juveniles altogether. And you bring up a good point. I mean, if we compare loggerhead and green sea turtle alleles to those of other species of sea turtle, like the hawksbill, which is critically endangered, there's probably going to be a very big difference. And I would expect there to be less MHC diversity because of bottlenecks and things like that.
Yeah, okay. Thank you.
Yeah.
So what, I guess, I know that you're kind of going towards in terms of the actual project, but in terms of maybe your actual personal goals, your academic goals, where do you see yourself going after this?
I've thought a lot about this. I mean, it gets brought up a lot in graduate school. I'm not opposed to working, you know, for a conservation organization, whether that's, you know, a nonprofit or even in the government sector. But I mean, I'm also, and I always have been interested in trying to give back to the types of institutions that are like Fairfield that afforded me some really wonderful opportunities. I feel very fortunate, and I have a lot of privilege that I was able to go on field courses multiple times to Brazil with you. And I really am attracted to small undergraduate institutions where the focus is undergraduate experience and teaching. You know, I mean, there's a lot to be said for trying to prep the next generation, and that's my overarching goal as of right now. I still have, you know, a number of years to kind of decide, and, you know, postdocs and things like that, but I would really like to be able to give back to an institution like Fairfield.
Yeah, that's great. Well,
Thank you for watching the Future Frogmen Conversation Series. Please check out our website at www.futurefrogmen.org.